A really good place to introduce the concept of genetic mutations is at the end of our discussion of gene expression. Genetic mutations are changes in DNA, and these are changes in DNA that ultimately can change the encoded protein. So I have a picture up here of a penguin carrying a mutation in the gene that encodes an enzyme for creating melanin. And in normal penguins, that allele encodes an enzyme that converts a precursor molecule to melanin. But in this penguin, that enzyme is defective. So the polypeptide that's made from the gene isn't functional, and this is then an albino penguin. So I've described mutation as a change in DNA nucleotide sequence. A change in DNA can occur by mistakes in DNA replication, or they can be induced by chemical or environmental mutagens, and mutagens are things that induce mutations or cause mutations. Now, the change in DNA may or may not cause a change in the protein structure or its function. If the change occurs in a region of a gene that encodes a protein, however, there are different types of consequences. So one thing I want to mention is mutations happen all the time. Our DNA is constantly taking hits, and there are multiple levels of repair. So not only is our DNA taking lots of hits, but we have lots of different repair mechanisms that come in and change those mistakes. In addition, most of our DNA actually doesn't encode proteins, and so most of the places where mutations occur don't affect the encoded proteins. Mutations are random. There isn't a more likely spot for a mutation to occur. They can occur anywhere in DNA. So let's look at the type of consequences that occur when a mutation occurs in a region of a gene that actually encodes a protein. So you've got a figure in your text that shows you an mRNA that encodes a polypeptide. And you've got a description of different types of mutations that can happen. And remember, these are happening just because mistakes happen in replication or because some kind of chemical or environmental mutagen actually damages the DNA. A nucleotide can be substituted, or a nucleotide could be deleted or added. Notice that a substitution can actually change one encoded amino acid, but certainly a deletion or an insertion would completely change the series of codons that are read, and so the encoded amino acids from the point of mutation. I want to go into a little more detail about this nucleotide substitutions. Here's an original coding strand of DNA. So the mRNA would be UAU, UGG, and so on. U's instead of T's. So UAU in the genetic code encodes tyrosine. Now, a silent mutation is one where there's a change in DNA that doesn't actually change the encoded amino acid. So UAC encodes tyrosine just like UAU. This is called a silent mutation. The change in the nucleotide does not change the encoded amino acid. And that's especially true if the change occurs in the third nucleotide of the codon, because the redundancy of the code allows for an amino acid to be encoded by multiple codons, and those codons differ by the very last nucleotide. A missense mutation is one where there's a change in the nucleotide, and that change actually changes the encoded amino acid. So in this case, UGG encodes tryptophan, but UGU encodes cysteine. So there was some kind of change that changed the DNA, a substitution mutation, and that created a missense in the protein. This is one that may or may not have a lot of consequence. This one did not, of course, because we didn't change the encoded amino acid, and so the polypeptide folded normally and functions normally. This one can cause a change depending on how large of a change the amino acid change is, and also depending on where the amino acid lies in the protein. Like, for example, if it was in an active site of an enzyme, it may be that every single amino acid that lines that active site is critical. This change then would significantly affect the function of the protein. But there are certainly places where amino acids aren't critical for protein's function, or if an amino acid substitution is conservative, like an acidic for an acidic or a charged for a charged amino acid, then the change might not be a big deal. A nonsense mutation is when the change in the nucleotide sequence changes a amino acid encoding codon to a stop codon. This is UGG encoding TRIP. A single change to UGA, or TGA in this coding strand, encodes a stop. That means that every amino acid after the point of change is no longer present, because remember, if the ribosome runs into a stop codon on the RNA, it stops translating. This, of course, is going to have a significant consequence, especially if it occurs early on in the protein. And then a frame shift is when we add or delete a nucleotide. Notice that that changes the series of amino acids from the point of insertion or deletion. Frame shift mutation is when a reading frame for the gene is changed, and that alters the sequence of encoded amino acids. A reading frame is the correct sequence of codons for a given gene. Every time the cell needs to make specific proteins, the same codons, a specific type of protein, the same codons are read and the same amino acids are used. Well, if in the gene there's a change so that the resulting mRNA is also carrying this insertion, that completely changes the resulting sequence of amino acids. This is an example of a really dramatic example of a missense mutation and the consequences. Sickle cell anemia is this inherited disorder where if you are carrying 2 copies of a mutated hemoglobin gene, you have these varying effects. So basically, someone with sickle cell anemia has a gene for the protein called hemoglobin, and hemoglobin is carrying oxygen in red blood cells. That protein is supposed to be a globular protein that makes red blood cells nice and fat and round and carry oxygen well. In sickle cell anemia, the gene carries a mutation, and the protein is no longer globular, instead it's filamentous, and it causes these cells to become angular, pointed, and sickle-shaped. The anemia part comes from the fact that these red blood cells get broken down, and there's a lot of clumping and clogging, causing damage to different tissues and organs. It can lead to heart failure, stroke, a host of effects. It's all because of a mutation in the protein called hemoglobin. This is the first 6 codons of the coding strand of the hemoglobin gene. The sixth amino acid, so actually it's 7 codons. The 6th amino acid should be an amino acid called glutamic acid. And glutamic acid has a negative charge, which means that it's going to interact with things that are positively charged when that little polypeptide is folding up to do its job. A single missense mutation that exists in the population changes an A to a T in the coding strand, so that a U is present instead of an A, AG versus GUG. Valine is a nonpolar amino acid, one that does not interact with anything charged. This one amino acid is so critical for the folding of the protein that changing it completely changes the shape of hemoglobin and the cells and causes all of these different effects of sickle cell anemia. Now, this mutation arose in Africa about 6,000 years ago, and the reason why it still persists today is, well, one, because we can treat sickle cell anemia, but also remember that we've talked about how you have 2 copies of every gene, 2 copies of every chromosome. Individuals that have only 1 bad copy yet have 1 good copy are actually resistant to malaria. And when this mutation arose, malaria was killing people, and the ability to have some sickled cells and some normal cells prevented the parasite from binding to all the red blood cells and destroying the red blood cells and killing the individual. So it's an interesting case where it's a single missense mutation that creates a huge effect, but it can be described as both beneficial because in a single copy it confers resistance to malaria, but also harmful because in double copy or double two versions that are bad causes sickle cell anemia. Now, I've been avoiding using some terms that have to do with inheritance, and those are the terms that we're going to talk about in the next module. We're going to hopefully tie together gene expression with patterns of inheritance.